Welcome to this episode of Mentors at Your Benchside, the podcast giving you advice, tips, and tools for getting the most out of your research. I'm Thomas Warwick, and today I'll be talking to you about different fixatives for flow cytometry. Fixation is routinely used in histology and cytology labs the world over as a way of keeping cells in stasis at a particular point to ensure that by the time they're examined, they've not deteriorated and the things you want to see are still there. This is also something that we often want to do in flow cytometry experiments. But there are several fixatives you can choose, and your sample type and downstream experiments impact your choice. Why should I fix my cells? There are many reasons why fixing your cells is a good idea. It will allow you to run the samples when you're ready, e.g. when a cytometer becomes available. This is particularly beneficial if you're doing a time course assay or paying for cytometer access. 2. You'll be safer because your cells are no longer alive. In some labs, fixed samples may be mandatory. 3. It's usually convenient and easy. And 4. If the antigen or target you want to look at is intracellular, you'll most likely need to fix your cells in order to permeabilize them, allowing the antibody/dye to reach its target. So what are your choices if you want or need to fix your samples? There are two commonly used classes of fixative: alcohols and aldehydes. Alcohols, usually ethanol or methanol, are precipitating or denaturing fixatives that coagulate proteins. They will dissolve lipids, meaning that they create large holes in the cellular membranes, including plasma membranes and nuclear membranes. Alcohols are generally good when you're interested in DNA analysis. Coagulation of the proteins around DNA allows even access to the dye, leading to a good coefficient of variation. But, as you may have thought, coagulation of proteins can be problematic if your antibody depends on a particular conformation of its target protein to recognize its epitope. So, in very many cases, alcohol fixation will mean you will be unable to detect your protein even though it's there in the cell. The exception to this will be small epitopes, such as phosphorylation-specific antibodies. Aldehydes are cross-linking fixatives and create bonds chiefly between lysine residues. In this way, they keep the structure of most proteins intact, which makes them an ideal candidate for antibody staining, as the epitope is more likely to remain intact. Formaldehyde. So, technically, the next two fixative examples are examples of the class of aldehydes. The usual aldehyde fixative in flow cytometry is formaldehyde, which will polymerize to become paraformaldehyde, or PFA, unless there's a small amount of methanol added to the solution. A common mistake is to say that paraformaldehyde is used to fix cells. In reality, paraformaldehyde is an insoluble white powder that won't fix anything. The solution we use is methanol-free formaldehyde. For fixation purposes, it should be diluted with phosphate buffered saline to a concentration of anywhere between 0.5 and 4%. Gluteraldehyde. Gluteraldehyde is extensively used in electron microscopy but is not as widespread in flow cytometry. This is because it's more effective at cross-linking than formaldehyde, making it harder for antibodies and dyes to stain structures effectively. It also increases cellular autofluorescence markedly. So, unless you have a particular reason for using gluteraldehyde, stick with formaldehyde or alcohol. A good rule of thumb. Bearing in mind what we just said, keep this in mind. Use formaldehyde for light microscopy and use gluteraldehyde for electron microscopy. How long will my cells last once fixed? You'll be pleased to hear that cells in ethanol are stable at 4 degrees or minus 20 degrees for months. For formaldehyde-fixed cells, that is reduced to around 1 or 2 weeks. Unlike ethanol-fixed cells, which can happily sit in ethanol all the time, we should spin cells out of the formaldehyde after 20 to 30 minutes of fixation, as prolonged storage in a fixative will increase autofluorescence. After this, they can safely be stored in phosphate-buffered saline. Increased autofluorescence is something to be wary of with all fixatives, and it can be particularly problematic if the target of interest is expressed only weakly. Fixation may also change the light scattering properties of the cells, which may be important to you. Combining fixatives, the best of both worlds. It is also possible to combine fixatives, and a common way of doing this is to first fix in formaldehyde and then post-fix in alcohol. The first fixative locks everything in place in the cells, and the second fixative makes larger holes in the cell, allowing easier access to antibodies and probes. It's also possible to use a permeabilizing agent, such as Triton X1000 or Tween 20 as a second step. Now that you know a bit more about the fixatives available, here are several tips to put them to good use. Do things in the correct order. The order in which you stain and fix may vary depending on the biological question being asked. Sometimes, if surface and intracellular epitopes are to be examined, it's best to stain surface antigens first and then fix them. But beware, some fluorochromes are sensitive to fixation. Fluorochromes such as phycoerythrin or PE or allophycoerythrin APC are large protein molecules and will be affected the same way as other proteins by fixation, so try to avoid alcohols. Small fluorochromes such as alexaflor 448 or FITC are generally unaffected by whichever fixative is being used. Use cold solutions. Fixation should generally be done using a cold solution as the fixation process is exothermic. The heat liberated could damage your sample and the antigens you want to detect. Be gentle. Cells should be vortexed gently while adding fixatives to ensure good fixative penetration and reduce cell clumping, and add at least 1 ml of the fixative solution to a cell pellet. So that's it for different fixatives for flow cytometry. Check out the episode description for links to related articles and resources, and don't forget to subscribe to the podcast to get more help and advice from mentors at your Benchside.